So in the previous lecture, we talked about middle ear anatomy and physiology, and we talked about the assessment of the middle ear using tympanometry and acoustical reflexes. So now we'll continue with anatomy physiology um, with the inner ear now. So the inner ear begins with the medial wall of the middle ear, from to which the stapes is attached to the oval window. The inner ear is where the end organs of the auditory system and the vestibular system are located. Just like other special organs, such as the eye and the tongue, um, the purpose of the auditory system is to actually convert an external physical stimulus, in this case acoustical energy, into a form that the nervous system can understand. So it can be conducted through the nerve, in this case auditory nerve, to higher structures where the acoustical signal can be interpreted as a sound. Um, and the language that the nervous system understands is electrochemical energy. So the ultimate purpose of the inner ear is to convert acoustical energy into electrochemical energy that can be carried by the nerves to eventually to the structure in the brain, the temporal lobe, where the sound is interpreted. And this process is what we call as auditory transduction. Auditory um, transduction, it refers to actually the transformation of one form of energy to another. And so here we're converting acoustical energy into electrochemical energy. And it's much like, ironically, the common analogy used to describe the inner ear is the microphones, which does the same purpose. They convert the acoustical sound energy into electrical energy uh, that's carried by the cables to an amplifier, and then it could be amplified by a speaker. But why I say ironical is because the microphones are actually based on how the inner ear or the auditory system works. But even with all the technological advances, we are still yet to construct a microphone that's as small, versatile, as sophisticated as our inner ear. The inner ear, even in the adults, is but the size of your pinky nail finger. So we're talking about a really small structure, which is highly sophisticated in terms of its architecture um, and um, and its mechanics. Something that I hope um, to explain in this few lectures. The inner ear is also known as a labyrinth because it's similar to um, kind of winding caves. Functionally, it's easier to study the inner ear anatomy as two parts, the bony labyrinth or the osseous labyrinth and the membranous labyrinth. Although anatomically the membranous labyrinth is actually located um, within the bony labyrinth. So the bony labyrinth is but a series of cavities kind of engraved uh, within a bone. And in fact, this bone is one of the hardest bone in our skull, namely the temporal bone. Specifically, it's a petrous part of the temporal bone. Um, and the membranous labyrinth, as the name goes, it's a membranous sac that kind of floats within this bony labyrinth. So here's in this illustration, you can see both the inner ears located in the kind of the bottom half of the skull. Um, just to orient yourself, this is the foramen magnum. It's the main opening of the skull through which the brainstem, the cervical vertebrae enter, um, and it connects to the brain. And here is an illustration of how small this whole inner ear is. It's but a 2 centimeter cube. Um, and what's fascinating is when you're looking at the embryological development of the inner ear is actually kind of adult size by in the beginning of the second trimester. So for all purposes, the inner ear is fully grown by the fourth month of gestation. And so it is true that the young embryo can actually hear sounds, although the sounds that they hear is probably muffled because it's traveling through all the liquid and the soft tissue around the womb. In this illustration, you're seeing the membranous labyrinth kind of taking the curves of the bony labyrinth, and it lies within the cavity. Here is an illustration of actually skull segments, and you can see that the inner ear is actually located as kind of series of cavities within the skull, within the temporal bone, and you can see the different shapes over there. So it's not like for instance the eye, where you can actually kind of remove the whole eye. Here we're talking about structures located deep within the bone, and that explains why it took us a long time even understanding the normal physiology of the inner ear, because even accessing the inner ear required kind of breaking into the skull, and that by itself results in an abnormal or it affects the normal functioning of the inner ear. And so we had to wait for technological advances to kind of to understand what's going on within the inner ear. And that also explains why some of our earlier theories about well, hearing actually were faulty or underestimating the function of the inner ear. It's another illustration of kind of a dried out part of the temporal bone. Here you're seeing those cavities. These are actually the scala of the cochlea, and we're going to be talking about that in just a bit, how there's actually three compartments within the cochlea arranged in a spiral fashion. Just to orient yourself again, this is a stapes of the middle ear cavity, and here you're actually seeing the porous mastoid bone and the mastoid air cells over there. So the bony labyrinth consists of three parts. We have the central kind of dome-shaped vestibule, just much like the vestibule of our home. And on either side we have, on one side we have the cochlea, the spiral cochlea, and on the other side we have the set of three semicircular canals. The bony labyrinth has a fluid known as a perilymph, and it's in this perilymph this membranous labyrinth actually kind of floats. One end of the semicircular canals has a swelling, and it's that swelling that we call as ampulla over here. Okay, here, so the vestibule is an oval cavity. It's located between the cochlea and the semicircular canals. And it's this vestibule that we have those two windows, namely the oval window and the round window. Again, to recollect, the oval window is where the foot of the stapes of the ossicles is attached to. And the purpose of the round window is actually to dissipate pressure when the stapes actually moves in and out as sound enters the inner ear. Again, in one end we have got the three semicircular canals, and an easier way of remembering them is SPL, superior, posterior, lateral. And they're roughly at right angles, or they're oriented 90 degrees to each other, and that serves a purpose that I'll get to in just a few minutes. It's in this vestibule and on the semicircular canals where the vestibular apparatus is located, which is important for balance perception. And again, in this juncture, just wanted to remind that The vestibular assessment is actually falls within the domain of audiologist. Okay. Uh, Number of audiologists are responsible for these vestibular assessments and diagnosing vestibular conditions. In fact, here in our program at ETSU, we have two vestibular specialists that are kind of world renowned for their research in vestibular assessments. Uh, Both of them, Dr. Faith Aiken and Dr. Owen Murnane, are affiliated with the VA. But that's an excellent opportunity that our students gain by being involved in this vestibular assessments. Okay, each semicircular canal, as I said, has one swelling at its end, and which is known as ampulla. And this is a significant anatomical point because that's where the vestibular end organs of the semicircular canals are located. The bony cochlea, um, commonly is referred to like a snail-shaped um, shell. It's also like the top of a frosty ice cream. Um, Earlier, it was believed that this coiling was to save space, and also coiling helps to kind of tuck in these sensitive organs and save them from damage. But now, actually, recently, research has shown that it probably has another important function that we'll touch in when we talk about the physiology. It forms the anterior part of the labyrinth, and it's almost horizontal. Horizontally, it sits on the vestibule. It's not a completely concrete bone. It's actually got holes at the bottom of the cochlea through which the eighth, the auditory vestibular nerve enters and kind of serve and reaches to the hair cells. So here you're seeing the bony cochlea in isolation. So you've got the modulus, which is that central core, and again, the central core, this is where the perforations are there through which the eighth auditory nerve enters and kind of spreads out to the different turns of the cochlea. In humans, the cochlea takes about 2 3/4 of a turn. In some mammals, the cochlea actually might have 4 or 5 turns, and it probably is what's responsible for the extended frequency range that many of these mammals can listen to. Again, it's about just half the size of the inner ear, and it's about only 1 centimeter wide, and it's only 5 millimeter from top to bottom. And it's easier to study the physiology of the cochlea as if it was a straightened out canal. So if you were to straighten it out, it would be about 30 millimeter in length. And it's such that it is broader for those basal turns, and it kind of gets narrower as you go to the apical or the top turns. So that was a bony labyrinth. The membranous labyrinth, as I said, refers to those membranous sacs that kind of float within the bony labyrinth. And they pretty much take the same shape of the bony labyrinth, except for the vestibule. While we have like one dome-shaped bony vestibule, actually inside there's two sacs. We've got the saccule, which is connected to the cochlea, and then you've got the utricle, over which the semicircular canals are sit. The membranous labyrinth is filled with a different type of fluid known as the endolymph. Okay, but the membranous labyrinth, just remember, floats in another fluid called the perilymph. Peri means perimeter, outside. Endo is inside, endolymph. And the composition, the chemical composition of these two fluids are different, and they play a role in the cochlear physiology that we'll be talking about. Here's an illustration of the membranous labyrinth kind of sculpted out of the bone. of an animal. So it kind of looks like a gelatinous uh, shrimp. And here you can see that the openings, the oval window and the round window. I've got a link over here for those who have the stomach for it. It's actually a dissection of the ear, so you might, well, hopefully it works, and you might enjoy watching actually a dissection of a cadaver ear. So again, the membranous labyrinth is those interconnected sacs, membranous sacs that lies within the bony labyrinth. Here again, you can see the uh, two compartment membranous vestibule. We've got the saccule over here connected to the cochlea, and then you've got the utricle over here that over which the semicircular canals sit on. And then you can see at the one end of the semicircular canals is that swelling known as ampulla. If you were to dissect the cochlea right at the center, if you were to dissect the cochlea right at the center, like almost like that, you would it would seem like that. So actually, the membranous labyrinth refers to that central compartment. Just because it lies within the bony labyrinth, it ends up dividing it the whole turn, each turn of the cochlea into three compartments um, over here. The three compartments are actually the top one is the scala vestibuli, the middle one is scala media center, and then the bottom one is the scala tympani. Here you're seeing the uh, eighth cranial nerve, the auditory nerve entering, and it's kind of reaching out into the different turns of the cochlea. So the membranous vestibule and the semicircular canals contain the end organs of the vestibular system. And as I mentioned earlier, the membranous vestibule has the utricle and the saccule. Um, and the utricle and the saccule have those hair cells um, that are actually what are responsible for capturing our head movement um, in any given plane, and it's sent through the vestibular nerve, and it's interpreted as a change in our head orientation. We, we wouldn't be talking too much about the vestibular system apart from this brief anatomy, uh, but just to give you an idea of what other structures are there in the inner ear. So the semicircular canals, as I said, we've got three of them oriented at right angles to each other, and they are there for a purpose because they are there to capture angular acceleration, which refers to our ability to perceive head rotation in different planes. Um, so now with three different semicircular canals oriented at right angles to each other, we can perceive head rotation on the three different planes. So whichever direction we turn, um, it stimulates one or more of the semicircular canals, and our brain interprets that as motion in a plane. Um, and it's almost like hair cells floating in a fluid, um, and they're deflected in one direction when we're moving in one plane. Um, and here's a link to an animation that kind of has a little bit more information about the function of the semicircular canals. So when you're turning a head, the hair cells, which are located within the semicircular canals, are displaced. And depending upon which plane we are moving, any given one or more of the semicircular canals are stimulated, and that helps us perceive in which plane we are moving in. The hair cells within the semicyclic canal are known as cupula. Again, the cupula are responsible for capturing angular acceleration. Then the membranous labyrinth in the vestibule, again, we talked about how that's divided into the utricle and the saccule. Um, the saccule is what's connected to the cochlea. It's connected by this narrow duct known as this ductus reunions. Both the utricle and the saccule are responsible for linear acceleration. So linear acceleration refers to our ability to perceive motion in a single plane, let's say up and down, or when you're moving in a car, like front and back. So that's different than the angular acceleration that's captured by the semicyclic canals, where that's the revolution of your head in any given plane. So the sensory regions of the utricle and saccule are known as the macula, um, while the sensory regions of the semicyclic canals that I mentioned earlier um, are located within the ampulla, that's swelling at one end of the semicyclic canal, and they are known as a cupula. So in this example, you're seeing that the macula kind of capture linear acceleration, like you moving front and back or up and down as if in an elevator. And when you're doing vestibular assessments, actually you try to target um, either semicyclic canals or the utricle saccule. You're assessing how well uh, these structures are capturing movement. So the membranous cochlea lies within this bony cochlea, kind of coiling around the central core or the modulus. It consists of three ducts or scala. So as mentioned earlier, we've got the scala vestibuli, the scala tympani, and between them lies the scala media. The scala media is also known as a cochlear duct, and it kind of floats in between the scala vestibuli on the top and the scala tympani in the bottom. So the scala vestibuli is separated by this from the scala media by this membrane known as a raisinous membrane, while the scala media is separated from the scala tympani, which lies in the bottom, by the basilar membrane. And specifically, this basilar membrane is an important membrane because it's over which lies the sensory hair cells of the cochlea. So here's an illustration. Here's a scanning image of a cross section of the cochlea. So if you were to take the cochlea turns and if you were to kind of make a cross section right over there and look into it, it's going to look like that. So here we're seeing the three scala, the scala vestibuli at the top. It's the larger of the three compartments. We've got the scala media over here that lies between the scala vestibuli and the scala tympani in the bottom. So it's this membrane that we call as a raisinous membrane, and the bottom one is the basilar membrane. So the basilar membrane is an important membrane because the organ of Corti, where the end organs are the sensitive hair cells, they lie on the top of the basilar membrane. And you can see the same in the other illustration also over here. So the scala tympani at the bottom, scala vestibuli at the top, and the scala media lying in between. So the oval window in the vestibule actually opens up into the scala vestibuli. So we've got actually the oval window right there, opening into the scala vestibuli, and the round window opens into the scala tympani over here. Um, so when the stapes moves in and out, is actually pushing the fluid. In this case, that would be the perilymph in and out of the scala vestibuli, and it's that fluid wave that moves around the cochlea till the top, and then it comes back all the way down and pushes out the round window. So the scala vestibuli communicates through to the middle ear by the round oval window, pardon me, and the scala tympani communicates to the middle ear by the round window. So again, the scala vestibuli and the scala tympani are filled by the perilymph, while the scala media that lies in between has this endolymph. Again, in this figure, we've got the scala vestibuli, the scala media, and the scala tympani at the bottom over here. At the very top of the cochlea is where actually the scala vestibuli communicates or opens into the scala tympani by this opening called as the helicotrema. However, the scala media that lies in between, it's kind of sealed from both the scala vestibuli and the scala tympani. So that's why the chemical composition of the endolymph that lies within here is different than the perilymph that lies in the scala vestibuli and tympani. So the scala vestibuli and the scala tympani is filled with the perilymph, while the scala media is filled with the endolymph. Here's another illustration of kind of the real cochlea. It's a scanning image where you're seeing the scala tympani, scala vestibuli, um, the scala tympani over here, um, and in between you're seeing the scala media. So it's this membrane that we call as a raisinous membrane. And this membrane is what we call as a basilar membrane. Part of my handwriting, the stylus is pretty bulky. So the basilar membrane separates the scala media from the scala tympani at the bottom, while the raisinous membrane separates the scala vestibuli from the scala media. And that's over here. Raisinous basilar membrane over here. At the top of the basilar membrane is where the organ of Corti lies. The organ of Corti has inner hair cells and outer hair cells where the final auditory transduction happens. Here's an illustration of actually the perilymph and the endolymph. Um, I believe this researcher kind of injected different colored rubber fluid, one through the oval window and one through the round window, I believe. Um, and then he cracked open the skull, and just by the number of turns over here, I'm assuming this is some animal like a rat or a gerbil, because we have more turns than the 2, 3/4 that we see in humans. But kind of it's a neat way of looking at the three different compartments, three different scala of the inner ear.